three-dimensional structure of a protein is determined by its amino acid sequence. The most important proof of this came from the experiments showing that denaturation of some proteins is reversible. So what is denaturation of protein? Let's first talk about protein denaturation. Protein structures have evolved to function in particular cellular environments. Condition changes result in loss of native three-dimensional structure of proteins, which in turn cause loss of function. This process is called denaturation. Proteins can be denatured by heat, extremes of pH, certain organic solvents such as alcohol or acetone, certain solutes such as urea and guanidine hydrochloride, or by detergents. These denaturing agents usually disrupt non-covalent interactions in a protein. In this case, no peptide bonds in the polypeptide chain are broken. Denaturation of a protein can result in exposure of hydrophobic surfaces and formation of protein aggregates. Therefore, denaturation often leads to protein precipitation. Certain denatured proteins will regain their native structure and their biological activity if returned to original conditions by removing the denaturing agents. This process is called renaturation. In the 1950s, the classic work of Christine Efferson on the denaturation and the renaturation of ribonuclease A revealed the relation between the amino acid sequence of a protein and its conformation. Ribonuclease A is a simple polypeptide chain consisting of 124 amino acid residues cross-linked by 4 disulfide bonds. Purified ribonuclease A denatures completely in a concentrated urea solution in the presence of beta-mercaptoacinol. Beta-mercaptoacinol is a reducing agent that cleaves the 4 disulfide bonds to yield H16 residues, and the urea disrupts the stabilizing hydrophobic effect, thus unfolding the entire polypeptide chain. Denaturation of ribonuclease is accompanied by a complete loss of catalytic activity. When the urea and the reducing agent are removed, the randomly coiled denatured ribonuclease spontaneously refolded into its correct tertiary structure, with full restoration of its catalytic activity. The refolding of ribonuclease is so accurate that the 4 intrachain disulfide bonds are reformed in the same positions in the renatured molecule as in the native ribonucleases. This experiment provided first evidence that the amino acid sequence of a polypeptide chain contains all the information required to fold the chain into its native three-dimensional structure. Subsequent work has shown that only a few proteins will fold spontaneously into their native form. Most of proteins require some assistance to fold into their native structure. Okay, in summary, the three-dimensional structure and the function of most proteins can be destroyed by denaturation, demonstrating a relationship between structure and function. Some denatured proteins can renature spontaneously to form biologically active protein, showing that tertiary structure is determined by amino acid sequence.